All right, so let's spend some more time talking about proto-oncogenes. What I'd like to do here is draw what the genes normally look like. So remember that, of course, we are diploid organisms, so we're going to have 2 copies of each proto-oncogene. And it's important that we have these copies, again, because they help drive the cell cycle, allow it to move forward, provide a promoter here as well. So these 2 are proto-oncogenes. And for now, I'm just going to put P-O for proto-oncogene. When it's time to move through the cell cycle to go from G1 to S, these genes are turned on. All right, so this is what we see under normal circumstances. The genes are kept off until it's ready to move through the cell cycle, and then the genes are turned on. So when they get mutated, these proto-oncogenes become oncogenes. And on the last whiteboard, I said when they become oncogenes, they're overexpressed. And we spent some time talking about that, so this is clear. So let's ask this question first. What happens if the genes, these proto-oncogenes, become non-functional? They're mutated in such a way that they're non-functional. They're not making the proper protein. If that was the case, they wouldn't move the cell cycle forward. And remember, for this to be linked to cancer, it's got to move the cell cycle faster, because cancer is characterized by rapid and uncontrolled growth. So if we mutated these 2 genes so that they were no longer functional, that would be the reverse of what cancer is. It would stop cell growth. So mutating proto-oncogenes to oncogenes cannot be the type of mutation that makes them non-functional. It has to be a mutation that makes them overactive. So let's ask the follow-up question here then. What happens if genes, I should say proto-oncogenes, become overexpressed? So remember, normally they're this way. So what happens if, just say one of these becomes active in such a way that its expression is much higher? If that happened, cell cycle progression would be faster. And this is what cancer is all about, faster, unregulated cell growth. Now, in the previous slide, I also said that only one of these genes has to be mutated. And I hope this explains why that's the case. In order to have the cell cycle move faster, all you really need to do is overexpress one of these alleles. It doesn't matter what this one's doing. This one can be doing the exact same expression level, no mutations at all. But as long as this copy here is making more transcripts and ultimately more proteins from this gene, that's all that you need. So let's write that down here. Only one allele of the 2 needs to be overexpressed. And if one of these 2 alleles becomes overexpressed, it becomes an oncogene and allows the cell cycle to move much faster. And we'll talk more about the difference between dominant and recessive in our next unit of material. But one of the features of dominant mutation is that you only require one of the 2 genes to be mutated. So because of this, we would say these mutations, going from proto-oncogenes to oncogenes, are dominant mutations. Only one of the 2 alleles is mutated. So the next question we want to tackle is, how is it that we can overexpress these genes? In general, we think of 3 different ways to make a proto-oncogene into an oncogene. And I'm going to throw a 4th one in as well. In this whiteboard, we're going to talk about the ways we take a proto-oncogene and make it into an oncogene. And the 3 ways I'm going to talk about are generating a missense mutation, gene amplification, translocation, like we talked about with the chromosomal mutations. And the last way is through viruses. Now, viruses themselves can cause translocations or gene amplifications, but I want to list it as a 4th way just so we make sure we talk about it as a way that proto-oncogenes become oncogenes. But I'm going to take them in order, so let's first talk about missense mutations. So remember, a missense mutation changes one amino acid. So let's draw a general protein that might be made from a proto-oncogene. It may make a protein that looks like this. And in this form here, it is inactive. And to become active, some stimulus occurs. And now that stimulus makes the proto-oncogene protein that's inactive here, it now makes it active. Folds a little differently now. It maybe binds this particular substrate. It does something so it is now active. So how is it we're going to go from a proto-oncogene to an oncogene simply by a missense mutation? So it becomes an oncogene due to this missense mutation by changing just this one amino acid. And what happens here is this mutation makes a protein that is always folded in the active form. So this protein is always active. It doesn't need the stimulus. Every time it's made, every time the gene is transcribed and it's translated, it makes this protein in a permanent active state. So I want to draw the 2 genes down here. We mentioned this before, but it's a point that I know students often struggle with. So I want to make sure it's clear. So here's our proto-oncogenes, and they're going to make this inactive protein that can now become active. But if it becomes mutated on just one allele, instead of making this initial protein here, it's always going to make from this one allele, it's always going to make the active protein, so it doesn't have to be activated by the stimulus. So as we said on the last whiteboard, all we need is one of these alleles to be mutated, because it only takes one to make this always active protein, and so now it's always driving the cell cycle forward. This protein, the inactive one that's supposed to be made, is doing its job, right? It is not driving cell cycle forward. But it doesn't matter, because this one has gone rogue. It is bypassing the need for regulation by this protein, and it's just driving that cell cycle. This protein can do nothing about it. Sad, but true. In editing this, I realized I made a slight error, so I want to correct it real quick. I said that the non-mutated proto-oncogene that is made is not driving the cell cycle. It would have been better for me to have said that it can only drive the cell cycle forward if the cell receives the proper stimulus. So it is still being regulated properly. The new oncogene, however, is not being regulated properly. So one is regulated, the other isn't. And that is why you only need one copy mutated. I'd like to spend just a little time talking about a specific example of a proto-oncogene to an oncogene called RAS. So normally in a cell, what you have are two proto-oncogenes called RAS. And what they do is they signal the cell to divide. And so what we're going to have on the outside of the cell here is a receptor. In fact, let's call it a growth factor receptor. And as long as it's not bound by a stimulus that tells the cell to divide, RAS is inactive. And more specifically, it's inactive because it's bound to something called GDP. Now, if we were to add a growth factor to this, the growth factor would bind to the receptor. So the growth factor binds to this receptor. And what that does is it triggers a response within the cell. And don't worry too much about the details here. I'll draw some of them here. But when that happens, this growth factor receptor here becomes phosphorylated. It will then phosphorylate a series of other proteins here. I could draw these over here, didn't need to, but I will. But they're inactive, they're just hanging out there. Growth factor binds, causes phosphorylation, it phosphorylates these targets, and ultimately what that will do is change the shape of our RAS in such a way that it releases the GDP and now binds GTP. And now RAS is active. Once it's active, it's going to phosphorylate a series of proteins that I'm just going to draw like this. We call this a kinase cascade because each of these are kind of a kinase that will phosphorylate each of these. And the last one that gets phosphorylated, what it will do is it will enter the nucleus. So let's draw the nucleus here. We should have drawn it here too, just to be consistent. And inside the nucleus, there is a gene that needs to be activated in order to drive the cell cycle. So this last phosphorylated kinase will now enter the nucleus and activate the expression of these genes necessary to drive the cell cycle. Over here, these genes are off because this protein, this activated protein, hasn't come into the cell because this activated protein hasn't been activated by RAS. So what happens when RAS becomes an oncogene? That is, what happens when one of these two copies down here becomes mutated? And now this one, this RAS is an oncogene. And I'm going to draw that cell here. This cell still has the growth factor receptor. It still has these targets that are going to become phosphorylated when the GF receptor is activated. But what's different here is the RAS protein is always active. You don't need the growth factor to bind here to activate these proteins, then to activate RAS. This RAS protein is always bound to GTP, or behaves as if it's always bound to GTP. And because of that, these proteins that I talked about in this cascade are always going to be phosphorylated. And it's always going to be active to move into the nucleus, to always express these genes necessary to move the cell cycle forward in an unregulated manner. Again, you can see here that it doesn't matter if we also happen to have in this membrane the proper RAS. The proper RAS is doing its job. It's waiting till it's activated by the growth factor before it signals the cascade. But the oncogene here, it doesn't care. It doesn't listen to that. It's always activating this process. And this one, this properly regulated RAS can do nothing about it. It's gone rogue, so to speak. Okay, let's talk about the second way that a proto-oncogene can become an oncogene. Remember, normally, and it's always going to be active to move into the nucleus, to always express that are properly regulated. Every time they're expressed with this promoter here, they make an equivalent amount of protein from each allele. And then these proteins go on to regulate the cell cycle. Now, when it becomes a proto-oncogene, what happens is, on one of these copies, the gene gets copied. Maybe just once, maybe multiple times. This allele down here is unaffected. Again, it only has to happen on one of the alleles. But now what's happening is every time you activate this proto-oncogene, this one makes 3 times the amount of proteins. This one down here still just makes 1 times the amount of proteins. But again, this one is driving the show, so to speak. It's making more proteins than is needed. And so this now becomes an oncogene. So it's allowing this cell cycle to move forward faster because there are going to be more proto-oncogene proteins made. Okay, the third way of making a proto-oncogene into an oncogene is by translocation. We'll talk more about this in class, but let me introduce it to you right now. And for this proto-oncogene, I'm going to give it a specific name, a very common proto-oncogene that becomes an oncogene to drive cancer. And I'm going to call it c-MYC. So these are both c-MYC, M-Y-C, proto-oncogenes. And then I'm going to draw over here a gene that is not a proto-oncogene. It's a gene that is constitutively on. That means always on. It could be like a glycolytic gene, it could be a housekeeping gene, some kind of gene that is just always expressed. Not necessarily at a high level, but always expressed. And we're just going to call this gene A for now. Both of these genes have their promoters that are specific to each of these specific genes, like so. Now, translocation can occur, and in this translocation, what's going to happen is this gene A here, that's always expressed, is going to move over here, and the proto-oncogene c-MYC is going to be translocated over here. The other copy is just fine. No translocation occurred. So c-MYC is still here on this other allele, and the A gene is still here on this allele. So how can this cause a problem? Well, remember, we said that this promoter up here is specific for A, and this promoter here is specific for c-MYC. This promoter is going to be highly regulated so that we only activate c-MYC when we want to move the cell cycle forward, when we want the cell cycle to go. So it's highly regulated. But what this translocation has done is it's moved c-MYC next to this promoter that is always expressed, because gene A's promoter is always expressed. So now this c-MYC is always going to be expressed. This copy over here, if it's not activated, is not going to be expressed. Again, it doesn't matter, because this one's going to drive the show. As long as one of these 2 alleles is overexpressed, the cell cycle will move forward. And so when c-MYC or other proto-oncogenes get moved in front of a promoter that is constitutively active, always expressed, it becomes an oncogene. So 2 key points that we keep talking about is that proto-oncogenes become oncogenes when the gene becomes always, when the gene is always active, and the change that has to occur only has to occur on one of the 2 alleles. Next, I want to just spend a little bit of time talking about viruses and how they can misexpress genes in such a way to drive the cell cycle. These virus genes are oncogenes. So let's draw a human cell here, and then let's draw a virus here. This virus is going to have an oncogene in it, and it's going to infect this cell. And when it infects this cell, it's going to have this oncogene protein here. This oncogene can do several things, but 2 general things it could do is it could activate a proto-oncogene. It takes a regularly expressed proto-oncogene, and when this viral oncogene is in the cell, it can activate this proto-oncogene, so that's always expressed. Alternatively, an oncogene from a virus could enter the cell and inhibit a tumor suppressor gene. And if a tumor suppressor gene is inhibited, the stop part of the cell cycle will be blocked. Both cases, whether the oncogene from the virus is this activating a proto-oncogene or inhibiting a tumor suppressor gene, will drive the cell cycle forward and can lead to cancer.